for the Wednesday research series. We are really honored to have Dr. Ramani Ramchandran. Dr. Ramani is a renowned scientist in the field of vascular biology. He received his bachelor and master degrees from the University of Mumbai and his PhD from Augusta University in Georgia. He was trained at esteemed laboratories at Augusta University and Harvard Medical School, where he developed his interest in vascular biology. He started his own lab at the NIH in 2002 and later joined the Medical College of Wisconsin, serving as a professor in the Department of Pediatrics and Clinical and Translational Science Institute. His research focuses on blood vessels patterning using human cell culture, zebrafish, and mouse models. He made a notable discovery about the role of cilia in brain vascular patterning, leading to its potential use as a biomarker in vascular health. With over 95 publications in leading journals and extensive NIH funding, Dr. Ramani has received several awards, including Distinguished Alumnus Award from the Augusta University. Today, he will share his work on cilia and its potential biomarker for vascular health. It's a great pleasure to have Dr. Ramani here, and the floor is yours. Thank you so much for coming.
Thank you, Dr. Mohammed, and thank you, Dr. Yeltsin, for this invitation. I'm particularly excited to be here today as we have signed the agreement between Medical College of Wisconsin and Qatar University. I see this as a new era, a new dawn of a relationship. And it really started with Dr. Yeltsin contacting me several years ago for a simple experiment that from his paper, we had contacted him. We wanted him to help us with an experiment. And he was so energetic, and he immediately helped us with that experiment. We published a paper, and that's how the whole relationship was born. What particularly excited me about working with Dr. Yeltsin was the fact that he kind of just committed himself to the purpose of the science, which I find very refreshing in a scientist. You know, he never asked me for authorship. He never asked me for anything. He just did it. And to me, that's the consummate scientist. That's what I like to see in a scientist. And I'm sure all of his trainees are like that as well. And so, I think that, you know, I see this as a beginning. And today, the way I've structured my talk is to show you the power of collaboration. You can do things by yourself, but I think the strengths are in the numbers, and that's kind of part of my title. Its strengths are in the numbers, and you need to reach out to people who have complementary expertise and who can then help you take the project to another level that you would have never imagined, whether it's an education project, whether it's a medical project, it's a clinical project, whatever the project might be. So I hope I will convince you that that's the vision that we should all aspire for. And feel free to interrupt me in my discussion. Although, you know, although it's a talk, but I don't want it to be too official. So, you know, feel free to kind of ask me questions and so on. I'm going to try to see if I can get rid of this disconnect thing here. Oh, it's gone. Okay. Thank you. So today I'll be talking about this very interesting. My first part of the talk is a little bit about the science that I do. I've kept it a little high level, not going too much deep into experimentation. And the second part of my talk is about this collaboration and what we will hopefully bring to the table together, and then we'll have a panel discussion that will arise from that. So I don't have any disclosure and financial, but I'd like to have some at some point. And so that's hopefully part of this long-term strategy as well. So we work on this very interesting organelle called cilia. It's singular is cilium and plural is cilia. And this is a structure that juts out of a cell. It's found in most cells in the body. And we are interested in endothelial cell, as shown by the picture there. It's like a hair-like organelle. It just comes right out of a cell. And it is both motile as well as non-motile. So it can move as well as not move. And it is made up of these things called microtubules. And these microtubules can go break up and down. And so it's a very dynamic structure. The structure can polymerize and depolymerize. And so it's very dynamic in a cellular structure. So as you can imagine, various disease conditions are affected by loss of cilia. Either ciliary genes or ciliary function. Here's a cartoon to show you pretty much every disease that you can think of in this slide, every organ, there is a ciliopathy that is associated with it. So our interest is in the brain, obviously, but and it just shows you the breadth and depth of the importance of cilium in biology. Not only is it important in diseases of the organs, but it's also important in cancer. And here's an example. Cells that are extremely drug resistant to chemotherapy and so on, they have longer cilium. And so people have thought that one could remove the cilium or make these cilium disappear and then make these cells sensitive to chemotherapy. So you can see that the amount of applications for this particular organelle is tremendous. And so our journey in the cilia biology really started out with our quest to look at cilium in the organism. And nobody had seen cilium in the brain vessels. And that was my area of interest. And we obviously, some people have already heard, I'm a zebrafish researcher, and the zebrafish is our model of choice. And you'll hear this marker constantly that we use in the field. It's a marker called ARL13B. It marks the cilium. Okay, it's a protein that's found in the cilium. It has functions outside of the cilium as well. So this particular protein is a membrane, is a member of a small GTVS family, and there is a genetic condition called Joubert syndrome where this particular protein is mutated, and they have deformed cilia and they have kidney cysts. So here's the structure of a cilium. So cilium has this hair-like, it's like a kind of a banana, actually. You can see the ciliary axoneme, which is the structure that juts out of the cell, and then it's attached to the membrane, and the ARL13B is found in the axoneme, and there are other proteins that are found in different parts of the cilium. So Dr. Brian Ceruna had developed a transgenic line from University of Toronto, where he had fused the ARL13B, the mouse ARL13B protein, to green fluorescent protein. So you could visualize cilium. So here's a classic example of a collaboration, right? So we contacted Dr. Ceruna, who was very kind enough to give us this line. And we crossed this line with a line that we had in the lab, where we had a endothelial cell, a specific expressing protein called M-cherry, which is red in color, and it was only in the endothelium because it was driven by a promoter, which is in the vasculature. So we had a kind of like hopefully a green cilium and a red endothelial cell. So that was the idea. Could we use these two particular transgenic lines to visualize the cilium in the brain vessels? And indeed we did. And so that's an example. That's exactly the picture that you see on the bottom right there. You see those green hair-like things coming out. Those are the cilium, and that's the red is the endothelial cells that are in the zebrafish head. Actually, that was a trunk, but it was the head vessel. So we further looked to see where else is this particular, how well is cilia expressed in embryonic vessels in the brain, and the same line was used, and here you can see that while the vessels are you can see in the panel A, the eye region, that's the area of the head. And you can see that the vessels are not yet formed, they're just assembling. While they're assembling, you can see the cilium is also being expressed in the endothelial cell. Why is that important? Because a lot of people thought that cilia is involved in blood flow. But at this particular point, there was no blood flow in the brain. So even before blood flow, cilium is already in the endothelial cells. So it may have additional functions. That was the implication of that. And here's a movie of a cilium actually with no flow moving inside the brain. So we can take all these kind of images. I'm sure you're familiar with that. So after flow, you can see the cilium in the lumen, and you can see the pictures here. the blown up picture, and the yellow is shown there. And you can actually see them move as you're taking different sort of snapshots of the vasculature in the brain. And you can also see them in the juvenile fish, actually as you get older in 33 hours, and as well as in 6.5 days fish. And what you can notice is that the cilium is like different sizes. You can see that they are long here, and they are found in the corners, and this is where flow is minimal. So it was known in human atherosclerosis that the flow at the corners of the vessels is actually quite low. And that's the area where cilia is enriched. Okay, so a lot of people said juvenile fish or adult fish don't have cilium. Well, clearly they do, but they are at the junctions. They're not in the main body of the vasculature. So this was the discovery that we had made in this paper, that previously nobody had seen cilia in the brain vessels. And using the transgenic technology, we were able to demonstrate that. So at this point, we wanted to know the significance of this. What is so important about this in the brain? This is where we contacted your esteemed colleague, Dr. Yeltsin from Qatar University. And he has devised, his paper was very elegantly done, where he had shown that he was able to quantify shear stress, which is, tangential force on the vasculature. And so we contacted him and said, hey, could you help us in calculating the shear stress in the vasculature in the brain, and we're going to manipulate it, and can you tell us what the numbers would be, and then we will tell you, what's the effect of that, sort of a thing. So his lab actually generated this data. So we take fish normally grow at 28 degrees, right? So when you just transfer them to 34.5 degrees, it's like a higher temperature. The heart starts to beat immensely, and then the blood flow gets to be higher. And he measured these things. This is the first time ever anybody has actually measured in a very precise manner the blood flow, velocity, pulse rate, shear stress in the brain vessels. It's never been done before. That was his contribution. So he measured all these things, and we could clearly demonstrate that when we induce heat to the fish, they do get a higher shear stress. So upon high shear stress, if you look at the cilium in the vasculature, so the top image is 28 degrees, and you can see all these green dots. Those are all cilium on the vessel. But if you look at them at 34.5 degrees, all of them have disappeared. So upon the induction of high shear stress, the cilium tend to disappear, okay? And they go away somewhere. And this observation is the first time that was demonstrated in vivo, but people knew about this in cell culture. Okay, that upon high shear stress, cilium just disappear. They don't know where they go. And this was confirmed in this paper. But what is the big deal? What happens to these fish if these things go away? It turns out they get hemorrhage. So the vessels become leaky. And you can see the picture there. That's the head of a 48-hour fish. And you can see blood pooling in the brain. Those asterisks are all blood that you see. And if you subject a wild type fish, a fish that has no genetic manipulation, you just increase the temperature, shear stress goes up, as I showed you, and the number of embryos that show bleeding also climbs up. As you can see, the numbers are around 10%. But if you have a background mutation on any cilia gene, and you do the same experiment, all these numbers go up. So essentially what it tells you is that cilia in the endothelial cells is somewhat important in the stabilizing the vasculature. So upon shear stress, the cilia goes away, the vasculature gets unstable, and things start to leak. That's the sort of the take-home message. So the hypothesis of this work was that if cilia is disassembled from endothelial cells upon laminar shear stress, as I showed you, can we detect these fragments in the supernatants from endothelial cells? So where are these things going? This is essentially the question. Are they like disappearing into outer space, or like, can we detect them? And so here we had a collaboration with a guy called Dr. Surya Nali, who's a world expert in cilia biology long before, and I started working on cilia. He's been working on cilia for like 15 years before me. And Surya is one of those great collaborators, just like Yeltsin, who basically, you pick up the phone and tell him, ask him something, and he'll just do it. And it was just great to work with him. So he subjected these epithelial and endothelial cells in an in vitro setting, subjected them to shear stress, and collected the supernatant and looked to see if he could see cilium or fragments of proteins. That was his experiment. So here are kidney cells. And you can see that the kidney cells, I don't know if you can appreciate it, but there's this box structure here where you can see that the cilium is translucent. It's this thin line here. That's 5 micrometer. When you induce shear stress, that thing goes away. And when you take the perfusate and put it on a slide, you can actually see all these fragments right here. Those are all ciliary fragments that are actually in the supernatant. Now, to make it more convincing, we stain them for cilia. So before shear stress, they are stained for a protein called gamma tubulin. This is green in color. After shear stress, those things are smaller in number because they're all gone. And we took these guys, these perfusates, and just did a Western blot for ciliary proteins. And you should, I remember I told you cilia is a microtubule structure, so it should not have actin in it. So you notice that the actin lane is empty on the isolated cilia, but the acylated tubulin is positive, suggesting that it is indeed a ciliary fragment, a ciliary protein. So that was epithelial cells. We looked at endothelial cells. The HUX also have these ciliary effluents and the ciliary proteins, but the cilia in endothelial cells are a lot smaller than the cilia in the epithelial cells. These are 5 micrometers. Those are just 1 and 2 micrometers. So they're a little hard to see sometimes, but we do see this concept. So now you might ask me, why do I care about deciliation? Who cares that it was deciliated? What does it matter? Well, if you can detect cilia in body fluids, okay, then it becomes a biomarker. For example, if you have a vasculature and you have a hypertension, right? And suddenly the hypertension is causing the cilia to get dismantled. If you can take a little blood and detect it, well, that becomes a surrogate marker for hypertension. So that is an absolute value in knowing this information. And as I just told you that these cilium are important for vascular stability in the brain. They're causing hemorrhages. So maybe you, in pediatric conditions like intraventricular hemorrhage, perhaps these structures can be restored in a way that we can prevent hemorrhages from occurring. And there are many cases. You know, you just finished watching the soccer World Cup, which was great in Qatar. We all enjoyed it. And you saw the concussions are such a big deal in those sports, in all sports, where there is a lot of physical contact. So you can see the brain injury is a very important place where cilia as a biomarker could be of high implications. So we started determining this by studying a disease called sickle cell disease. So sickle cell disease, I don't know if you're very familiar with it. These patients have hemorrhagic stroke when they are very young. So at 20 years of age, they get a hemorrhagic stroke. So they get a lot of bleeding in the brain, and they get a lot of pain, episodes of pain and crises. And their red blood cells are sickle in shape. So they clog up the blood vessels, and they don't carry oxygen very well. And so our hypothesis was these red blood cells will hit the cilium because they're hitting the blood vessel wall and collect the cilium. And that's how they remove the cilium physically from the vasculature. And maybe that's why these people are getting these hemorrhagic strokes and they are getting unstable vasculature. That was the hypothesis. So a second collaboration with Dr. Zanotti from Duke University. I don't even know Dr. Zanotti. I just picked up the phone and said, you know, you're a world expert in sickle RBCs. Could you help me do this experiment? And she said, what do you want me to do? And I said, can you throw a few red blood cells from human patient on a bed of endothelial cells and see if it sticks to them? And can you see cilium? She said, sure, I'm happy to do it. So this is her movie. She shows you a dynamic circulation of red blood cells inside a mouse artery. And you can see them all clogging up. All those very intense, bright structures are all red blood cells. They're clogging up the vasculature. So the importance of that is, can red blood cells in blood interact with cilia on endothelial cells? What is the implication of that? They would remove the cilia or cause ciliary dysfunction. They could cause blood vessel occlusion, or they could change the viscosity of blood and change the flow rates in the blood. So Rahima's lab then did the CLEAR experiment. She took the human red blood cells from patients, mind you, and she put it on a bed of endothelium under high shear stress, and brain endothelium in this case, and then asked how many endothelial cells stuck to how many red blood cells stuck to the brain endothelium, and what is the percentage of cilia before they went on flow and after they came out of flow. So here's the data. So the sickle cell RBCs, so RBCs from patients who have sickle cell disease, they have very high rates of sticking to the endothelium. That's 60%. Most of them stick very like a glue to the endothelium before they are put on this assay. We just did a flow cytometry for the cilium on the RBCs, and we noticed that sickle cell RBCs already have 20% cilia expression on them, which was a surprise. We didn't think they would have any cilium on them. They already have some ciliary protein expression on them, and after flow, that number, 20%, went up to 65%. So clearly, the additional cilium was being captured once they interacted with the endothelium. So our hypothesis seemed to be quite accurate in that sickle cell RBCs adhere to brain endothelial cells, collect the cilium, and then perhaps exit and so on. So now you can imagine if we could take red blood cells from a sickle cell patient, and we could now detect cilium on them, and if their numbers went up over a certain number, maybe we could predict when they come to the clinic every 6 months when they will have an episode of seizure and pain. So now you have immediately made an impact in the clinic. This is direct human clinical data. This is not mouse RBCs or anything. It's all human data. So you can again see the power of collaboration. I don't have sickle cell patients in Wisconsin. She works in Duke University, which is in the southeast corner of the United States, where there is a high proportion of sickle cell disease. So again, the power of collaboration is immense. So when we looked at human RBCs, you can see this picture here, and here are all these RBCs have some speckles of green, which is the ciliary protein. And when you look at mouse red blood cells, you can actually see the ciliary protein sticking on these RBCs. Those green stains are markers of ciliary protein, and you can actually see, and they're being quantified on the right. So these RBCs contain these ciliary fragments. They're sticking to them. So you could take RBCs from a FACS sorting, and you can actually quantify them. That's an assay. And in fact, I'm in the process of forming a company on this concept. And so we are now determining if cilia can be a biomarker of vascular health. So when you took these patients, the sickle cell RBCs, plasma from these patients, and we looked to see if there is more proteins in their blood. Because when these RBCs normally lyse very easily, so we hypothesized if they carry all these cilium, they must have more ciliary proteins in their plasma. And indeed, that's the case. We looked at 3 different ciliary proteins. All of them are higher in and the ciliary proteins are higher in the sickle cell blood than in the normal blood. And if you look at the ciliary structure, these proteins that we detected are from different parts of the cilium, meaning the cilia is getting shredded. Basically, you can find all these proteins at different structures of the cilium in the patients. So we feel that this is a very powerful concept, and we have now determined that the mechanism of deciliation is oxidative stress. I'm not going to show you data on that. So oxidative stress, when the sickle red blood cells bind to the endothelial cell cilia, the reactive oxygen species goes up, and that causes deciliation. I mean, we can restore the we can quench the reactive oxygen species, we can actually get cilia restored. So I'm not showing you that data. So the summary that I'm telling you so far is that when you increase shear stress, you can cause deciliation and reduce the expression of cilia in these endothelial cells and epithelial cells. I showed this in vivo, and in clinical conditions such as sickle cell disease, I already showed you that sickle RBCs, both in mouse and human, carry cilia post-interaction with brain endothelial cells. And in plasma, I showed you enrichment of these proteins in these sickle cell blood. So this paper was published in JCI Impact, which is Insight, which is a high impact publication. And here was Dr. Yeltsin, Dr. Nally, Dr. Janati, Dr. Palachik. All these people contributed to this study. Without this contribution, this paper would not have been accepted in a top journal. I made a podcast on this. If anybody's interested and you have some free time, it's 20 minutes of your life. You can go and listen to it on when you're driving to work or whatever. But the point, again, this is another way of disseminating science. We have to disseminate science to the general public. People need to know what we are doing, why we are doing, what's the importance of all this. And so I'm very active in that sphere as well. So some of the unpublished data that now I'm collecting is what is its role in other conditions. So Dr. Rarick is a cerebrovascular physiologist. Again, a collaboration where he has a traumatic brain injury model in mice, and it's a very common problem. There's 1.7 million cases per year in the United States. So 3.9 million cases of mild TBI, and it's a very devastating condition. And these guys have short-term and long-term functional deficits, and the quality of life can be really, really poor. And biomarkers are needed for this. So basically, he has done a cortical impact model where you impact the brain, and then look at, and here's the brain where the impact has occurred. It's like mimicking some shrapnel wound or something like a soccer ball or something. So you can see that the right side has got bleeding and the left side of the brain is not. And he collected plasma, and we looked to see, does these ciliary proteins go up? And in fact, in one day, the ciliary proteins are already up at day 1. And by day 7, the ciliary proteins are already down. And we can actually see that on day 7, you can actually see the wound is healing. So when the wound is, you can see that very bloody at day 1, the ciliary protein is high, and it correlates to the extent of injury. So it's amazing that so it's not only the severity of injury, but also recovery. So you can imagine if we had a biomarker that told people that when they could go back to work, it would be huge for these patients. So we found another protein, ciliary protein, also doing the same thing, slightly different patterns. So the point of this is that plasma samples from TBI rats show enhanced ciliary expression at early as day 1, and maybe correlates with the disease severity. Implications of future. Clearly, can cilia numbers and RBCs be used to determine underlying state of the endothelium? Other conditions when flow is influenced, preeclampsia, atherosclerosis, name it. Maybe in all these conditions, cilia could be used as a biomarker for various things. So we are working on that, and do specific signatures of ciliary proteins exist in blood that will tell us what is wrong with in these patients. So these are questions that we are developing. So this is the group that worked on it, and all the universities that participated in it. And you can see we have funding from the NIH. So really a good example of how a collaboration resulted in a product that is now going to hopefully become an impactful in the clinic. So we are very proud of this. And so I'll switch gears from here to show you a little bit about our university and our sort of vision of how we see this collaboration with Qatar University developing. So this is our campus in Milwaukee, and that's my lab right there in that building, where I have put a small little rectangle or square around. And it's a sprawling campus. We have lots of hospitals, we have children's hospital, adult hospital, we have biomedical research centers. We have it's a very sprawling enterprise. And so Medical College of Wisconsin, its primary medical school is in Milwaukee. We have 2 satellite centers in Green Bay and in central Wisconsin. We have 26,000 employees, $300 million worth of annual research spending, $5 billion in combined revenues. And I don't want to tell you all the numbers. The point of this is to show you that we are very active in our community, and we have various other organizations that are part of this. So our Office of Global Health or of community is got this vision. We got established in 2020 of January. They want to be a leader in global health that promotes equity across neighborhoods and nations. And we want to really focus on multidisciplinary collaboration that integrates and advances global health science. So this is our major theme: community engagement, education, patient care, and research. We talked about this already in our meetings, and this is what we want to do. Some of you have already interacted with this office. Tiffany Fraser is the person who's been in touch with us. Dr. Cassidy is the associate dean of this enterprise. Dr. Stephen Hargarden was the founding dean, and Melissa is a recent joint coordinator of this position. So our guidelines, our ethical guiding principles are essentially partnership, bidirectional input and learning, We want to empower the host country and the community to define their needs and activities, sustainable programs and capacity building, compliance with laws, ethics and standards and code of conduct, humility, cultural sensitivity and respect for all involved, and accountability for actions. So that's kind of what our guiding principles are. This is just shown, we endorse the brochure declaration principles for promoting equitable global engagement. And these are very much the same thing that I just read, but presented in a different way. So the take-home message of my presentation is that we're all in this together. And the strengths in numbers, not only within the United States, but also across the globe, and collaboration is the way to go. And so let's build on the existing collaboration between MCW and QU. And it could be at the education level, it could be the research level. And any one of you in this room who wants to participate, you should just contact, take the initiative, talk to any one of us, and we'll make it happen. So we want ambassadors. We want people to be passionate about this. And I'm going to leave you with a video that sort of summarizes what I'm trying to do with this sort of in my sphere of things and my collaboration. So thank you for your time. I'm happy to answer any questions you may have.
Okay, maybe I can start.
Yeah.
So, have you detected the levels of cilia in the normal, not under shear stress?
Yes, we have.
But are they comparable between different, kind of ethnic groups or between genders? And I think that this is all, yeah.
It's something that we will collaborate. In fact, we think that even if we exercise, for example, blood change.
So you think the next obvious step is to start comparing different disease cohorts compared to match controls?
And with male, female, race, sex, all sorts of
And these are free, or they're all bound to RBCs?
I don't think that they're definitely some are free. Yeah, it's a very good question. So, you're really getting at the heart of specificity. So, cells have a mechanism where they secrete but the cilia very different. And cilia does not it doesn't have ribosomes. So therefore, any protein that is in the cilium is from the cell that it resides in. So we think that by determining the signature of the various ciliary extracellular vesicle secretions from the different cells, we would have a profile. And that profile will be matched by algorithm or some way to what's been seen in the blood. And by that, we would know what the origin is. That's the long-term vision. So the short-term vision, I don't know at this point. Yes, correct. Yeah.
We noticed on your images of zebrafish under high temperature, when the cilia is removed, there still maybe the basal body is still there. Do these cilia grow again?
Yes, they do. They grow back in half an hour. So our thinking is that regeneration of cilium is a natural process, but as we age, that process gets diminished. And that's probably one of the reasons why we have diseases that are emerging in various settings.
Thank you.
So when you talk about the oxidative stress being the mechanism of detachment of the cilia, could it be just the actual shear stress? Because obviously you have this the actual pressure of the blood is changing, fluctuating, especially at areas of bifurcation of vessels. So could it be just physical?
It could be. So we don't know whether it's a chicken or egg problem, whether the altered shear stress is causing oxidative stress to force deciliation, or is it the red blood cell interaction that's in. So it could be both. Yes, correct. You're right.
Dr. Ramani, the presence of cilia in blood, is the chronic thing, or like after some injury it goes up then it goes away, or somehow it's
So I don't have a definite answer for that question, but except I can say that in the preclinical models of traumatic brain injury, clearly when the injury occurs, when the hemorrhage occurs, they go up dramatically. And then as the injury is resolving, we're starting to see a decline in the number. So I think that hopefully when we get the human data in human traumatic brain injury, that will replicate. So yes, I think that it's a dynamic process, and that not only the injury state of the vasculature will be replicated by higher ciliary numbers, but the resolution of the vasculature injury will be associated with decreased cilia numbers over time.
Hi. So for the flow cytometer analysis, you mentioned that there is an increase of cilia on blood cells. So is that attached to the blood cells themselves? And if so, what is the mechanism of the cilia attaching to the blood cells?
It's a great question. It's a great question. We don't know the attachment, but we predict that the ciliary structures have lots of receptors. They have G protein coupled receptors, they have integrins and so on. So it wouldn't, and red cell membrane has lots of integrins as well. So it wouldn't be surprising that might be one source of it, right? It could be cell adhesion molecules. We don't know any of that, and that's part of our grant to determine that. So that's what Rahima is doing. Yeah.
So going back to Dr. Hassan's question, I think it's very interesting what you find with the cilia protein 1 and 2. The fact that after one of them goes down after recovery and the other one goes up. And it will be very interesting to see if this replicates in humans, and whether the ratio between the two at some point can actually tell you the level of recovery.
Exactly. So we don't believe that all ciliary proteins are going to have the same profile. And so this is where one has to sort of work on this a little bit more in detail. But the surprising thing for us was that RBCs were a source of cilia to begin with, because cilia is originating from the nucleus, and RBCs don't have nucleus. So the only way they could be collecting the cilium is scavenging and so on. So to me, it's a bit of a surprise, but I think that's where the opportunity is from a detection perspective. So.
No further question. We might as well start the.
Yeah, sure.
panel discussion. We'll make it brief. Hopefully we'll get some questions also from the audience. So if I might call Dr. Mahan Asma, Dr. Hussein Yeltsin, and our grad student, Mahmoud. Okay, so if I may start with Dr. Mahan. So as you all know, we just signed an MOU with the University of Wisconsin Medical College, and this comes after successful series of MOUs with RCSI and Alberta, McGill. So in terms of what do you think the value of these kind of international collaborations? What are the benefits from having such international collaborations between Qatar University and such a prestigious university such as Wisconsin?
Well, you can hear me, yes. I believe that international collaboration is a complex process. It's not, sometimes it's frustrating. It's not easy to have scientists from the same institute working together, let alone having scientists from different institutions working together on the same project. Despite that, and despite the challenge, I believe the benefits of international collaboration is worth every effort. When scientists come together and work on a one project, they can make a breakthrough achievement. They can publish a high impact paper. Like one scientist will provide the samples, the other will provide the equipment, the third will provide the student. So the benefits are tremendous. And I believe that QU is trying to attract such international collaboration through these MOU agreements, and also through funding and through grants. In 2018, they launched the International Research Collaboration Co-Fund, the IRCC, and they also we had this, another call with Japan, the Japan-Qatar Collaboration Fund. So I believe Qatar University is going to the right path, and I think we have good examples also. We have experience.
Actually, your talk was a clear manifestation of the impact of such collaboration. So if I may actually ask Dr. Hussein, what do you think the challenges that such collaboration would actually face, and what are the proposed solutions for such challenges?
Yeah. So I think as Dr. Rahmani's talk, it gave a good example of a scientific collaboration, and of course it involves lots of challenges, but also opportunities. And I would like to explain maybe the challenges we had in this example, and maybe we can generalize from there. So there's a good story behind this whole collaboration. I thank Dr. Rahmani for the nice words and like support. He's our role model, actually. He's a very good example of a very nice, good scientist, supportive scientist, open. Anyway, so a few years ago, it all started with an email from Dr. Rahmani's lab. And like they were working on this cilia model, but they had a challenge to measure the flow. But it also includes some chance, some luck. That student was telling me, Asa, now after that we became friends. They put the animals into high temperature than they are used to, but by chance, with the wrong incubator, and they realized the brains start to bleed. But as a good scientist, they said that these two might be related. They didn't just this is a wrong experiment, we will repeat. They had a clue in there, and they want to explore more. So this luck gave them some chance. But then curiosity, scientific curiosity was there. So they want to understand the reason. Then they contacted us. I received this email. Then I phoned back. We talked, and we made a Zoom call. It just take maybe 2 days. Then we designed the experiment, and did it fast. We didn't know each other. Then Dr. Ramani led our group. He guided us how to run the experiment, what to do, and like design my team involved, Mahmoud, Fatih, other members of the team. Then we finished the experiment in a short term and published the paper. So the challenge was we didn't know each other. Like this was different universities, different set of skills. But like the opportunity challenge was there. We take it in a good manner. We were open to collaboration. Like the both teams, they were very supportive. So even though the challenge was there, like our difference was there, it was a challenge also. Like but we took it as a advantage. I mean, they were working 8 hours before, but we were using 24 hours because we were communicating at the end of the day. They were sending us email, we were responding a few hours later. So all of them worked out well. I mean, it just depends on where you look at the problem challenge. So it's good to see that this initial email came up to this memorandum of understanding at the end, which will help to our students and hopefully researchers from both sides, from their side, from our side. That's all I can say.
Fantastic,
Thank
very
you very
nice,
much.
very inspiring. If I may ask, Mahmoud, since you're a living example of such collaboration from a student perspective, how do you describe, having such opportunity to work collaboratively with external partners?
Yes. First of all, I think international collaborations are very good for us. I'm happy to see this paradigm shift in incorporating more undergraduate students into research in the past years. This publication is very dear to me because it was my first publication, and I was happy because we got positive results. Sometimes negative results is also results, but it feels good and gives a huge motivation and push when your first result is a good result. So I think it shapes your personality as a scientist when you start interacting with people like Dr. Ramachandran from all around the world. 15 hours difference between us by the direct plane. You interact with older people in your lab, such as master students, PhD students, postdoc like Dr. Fatiha, who was working with us on this project. And lastly, I think it's good to see that the motivational rewards, whether it's a physical reward or moral reward. For example, we received financial rewards for this publication. The university did an excellent job and highlighted our work. And also, when you see your name, this first publication, you see, okay, I will see my grandchildren. If the internet is there, they're going to see that their grandfather did some science work. We hope that we, through this accumulative work, we leave a little bit of impact for science. Also, like I would like to thank Dr. Hussein very, very much. He has been a role model for me. I worked with him in his lab for 5 years. He's, as Dr. Ramani described him, is a real scientist who's energetic, and he's like an invisible soldier. But those who were close to him, they know very well what person is he. Thank you.
Actually, second that. And thank you so much for sharing your experience. And really fascinating. So, leaving the best to the last, Dr. Ramani, how do you see? This is just to wake everybody up in case someone is sleeping. So, Dr. Ramani, how do you see ways forward? Now, we just had a chat just before the talk. There's so many ways to collaborate. Dr. Raha mentioned the IRCC grant, which is joint grant, between our two institutions. Talk about education, training, exchange of students. How do you see moving forward?
Okay, yeah, so I'll address that. But before I address that, I want to say that with collaborations, one of the key ingredients of collaboration is trust. And people in the scientific world are too worried sometimes about who gets credit, whose research is going to get scooped, and whose XYZ is not going, and whatever domains they have. It's just all these silos are really what we need to break down. And I think the COVID is a really good example. When COVID came into this planet, within 16 months, there was this amazing collaboration between industry, government, scientists all over the world. We solved the problem. I can't prove any better than that. That convinced, even today, NIH has changed the model of how diseases will be targeted, because we have not been as successful as we have been with COVID. So that's the example. So it's all about collaborations, multi-institutional, multinational, and so on. But specifically about this collaboration, so the question that you asked, I think the first thing I see is there are three pillars that I can immediately see that we're going to make an impact on. First one, we talk about education. So I think medical students in Qatar University will have an opportunity to go to MCW to do summer research. MCW medical students will come here to do some summer experiences. Perhaps there'll be clinical shadowing. I don't know. Those things have to be worked out, the details. So medical students will be one of the educational sort of pillars. Graduate students could be the second in that education pillar. They could be students here who could have a mentor at MCW as their co-PI, co-mentors, student from MCW, could have somebody from here on their committee and evaluate. So I see education in these two students. And as we get success in these two, then we build other student populations. I know the Dean of Pharmacy School is right in front of me, and perhaps pharmacy would be another avenue to tackle. So education is clearly, I think, is something that we should really focus on. The second area that I think we can also immediately focus on is team-based research teams, sort of research. So for example, there could be a select 3 themes. Let's say, I'm just making up right now, not that this has to be the themes. Cardiovascular, engineering, precision medicine, something like that. Then we would essentially have interested faculty from QU as well as MCW participate in those themes. And they would have Zoom calls and so on and so forth, or however forum they want to dictate. And then we would ask them to come up with common projects that within 6 months, we would like to see a proposal, an aims page, a grant. And then we would use that to support it. And then within 12 months of that, we would expect a publication. We would expect a grant, something to come out of it. To me, so each of these groups will have perhaps some co-leaders who will be responsible for making sure the group functions, and they would be accountability. Like, we would talk to them and ask them, what are the obstacles that you're facing? We want to remove them. So that would be on the research side of things. The third area that I can come up with is clinical trials or clinical avenue, where we are very good at conducting clinical trials. That's something that you has interest in. We could train them in that. We also are very good at rare diseases, in precision medicine, sequencing, and so those kind of things. And so that's, I think, one of the first things that struck me about QU when I first came was that there was this repository of DNA and blood from every single person in Qatar. That's an unprecedented opportunity to do precision medicine the right way. And so to me, Medical College of Wisconsin would greatly benefit by partnering in the precision medicine space as well. And so I think these are, and I hope I gave you guys specific examples. I'm all into specifics. I'm not into generalities. Because the more specific you are, the more the programs get developed. Now, Doesn't mean that these are the only things. So don't get, don't take that as home with you. These are starting points that I just came up with in this discussion forum. It may be 3 different themes as they emerge, the talks might emerge. But we want continuing sort of activities and that could culminate in a symposia here, a symposia there, an alternate symposia. To me, sky's the limit. If you want to be innovative and come up with an idea and you want to bring it to us, we could make it happen. It is not about me wanting this or Yeltsin wanting this. It is you wanting it and them wanting it. I mean, the respective faculty, students, and fellows and so on. It's not, we want it for sure, but I want you to own it.
Thank you so much, Dr. Mani, for the question. Dr. Firas.
Thank you so much, Dr. Han, for joining us and the panelists as well. When you start dealing with collaboration, it's definitely fantastic and fascinating. We all go through this experience, and we feel happy when we get those publications in high impact journal. I just had feeling of like when I have an idea, like now you have a model, something authority in a field, and you start sharing this with so many others. If something is going to be an IP, coming out of this, doesn't make it complicated. You can go for a service easy, but now you are sharing with six universities. Maybe they're going to claim some IP in whatever you do. How this complicate things? We are pro collaboration, but how we also worry about this IP thing?
Very, very legitimate question. So I think the intellectual property is definitely an area that actually is ripe for collaboration. So, people often, and I like to see things as we need to share. It's not about, for example, this project is a good example. Now, there is intellectual property that is coming out of this. There are these universities that are participating in it. But the universities are not going to directly get the intellectual property because the original patent's coming from Medical College of Wisconsin. However, when I license it to my company, which I'm forming, I plan to, all the people who help me in this, they're going to participate in the company in some ways. So what I'm saying is we are developing the blueprint. We don't know. I would be lying if I told you I know how to resolve that issue. However, I do believe if we show a model in which sharing of the royalties is beneficial to 2 parties, then I think there is strength in numbers again. So I think each institution has to look at it from a long-term perspective rather than a short-term gain. And hopefully my job is to convince people that it works. And so yes, it's a hairy topic, but it's a definitely an area of opportunity. For example, I'll give you a concrete example. Now I'm not going to be able to develop this by myself. I'm going to find a bioengineer to do it. You think a bioengineer is going to do it for free? I got to give him something to do this. And so at that point, at that negotiation, that agreement would occur, and that automatically the person would get a piece of the action. So I think one thing I'm touching on all of this here is that we have to be more giving, whether you are a top-ranked university or a bottom-ranked university. You still need to be giving. And I think giving is where you get more. I think that's my philosophy, and I'm hoping that it will translate into concrete products and it'll be an example.
Actually, if I comment on this, I think because one thing we were discussing, or you ended up with, is the how to sustain, this kind of collaboration. We have lots of MOUs, but do these MOUs, help us in any way if nobody's using them? So I think in order to make the recipe successful, it's good to start with mutual understanding. So even though the MOU states, collaboration, education, exchange with students, it's very good to define in case of, such, collaborative project, in what kind of royalties there will be, because then everybody will start on a solid ground and there will be no area for confusion. I think this is very important for sustainability for both parties to be happy. That's fine.
I completely agree with you. I think, but it will require a mindset change. And unfortunately, when the business of medicine intersects with academic activities, often the business seems to trump the academics. And how do we prevent that from happening? It's a challenge, but I think with the right set of leadership, it may be possible.
It's a challenge. Any more questions, inputs from audience? Okay, no more contributions. Thank you so much, Rahman. It's really an honor, pleasure to have you here. We welcome you always to Qatar University, second home.